We were just in the middle of stimulating neural activity, that kind of, or that basic category of methodology. So really, I had just started talking about different ways to stimulate neural activity. I think I really briefly talked about transcranial magnetic stimulation and the deep brain stimulation, and then gave you the short video about optogenetics. Some of the really early work on this kind of electrical stimulation of the brain started out with this single cell kind of stimulation. Wilder Penfield was the person who originally gave us that mapping of the homunculus in the primary somatosensory cortex. So remember, our primary somatosensory cortex, where we are feeling our body, is up in our parietal lobes, and he would go in there. So this would be people who were, I think it was usually that they were getting ready for some brain surgery, maybe for epilepsy or something, and their scalp would be open, and since there aren't really pain receptors in the brain itself, they could go in and stimulate single cells. And so he stimulated these specific areas, and let's say it's somewhere sort of in the dorsal area, but up towards the top, and they would say, oh, I feel a little tickle in my knee. And then you move a little more laterally, and I go, oh, I feel a tickle in my, as we go farther and farther into my arm or my hand or my thumb. And then as we move more ventrally, a little bit down in the primary somatosensory cortex, I feel a tickle on my lips. And so he mapped out this primary somatosensory cortex. He similarly mapped out our primary motor cortex. Another example is using this kind of single neural stimulation for stimulating the visual cortex. And what that does is produces these flashing points of light, or that's what people report. And note that these are not recognizable objects, but just these kind of flashing points. Probably, my guess is they would be little features like a little vertical bar, but very small because it's a single cell in the visual cortex. Moving on to recording brain activity, I'm going to start with electroencephalography, or EEG, and start out with this idea that really our bodies, our brains, are constantly emitting these electrical signals. And even though the bones and tissue are blocking a lot of this activity, as we get out, especially as we get outside of the skull, some of the current is going to reach outside of the body. And we're going to see that different frequencies in different locations that we can read along the scalp, we're going to see those as being associated with different mental states and different operations, different tasks that we do. And my favorite picture to talk from is maybe of all time is this looking at electrical activity over the brain and these different stages of sleep. The electroencephalograph was invented by Hans Berger in 1929. Monitors electrical activity by putting these electrodes, or kind of attaching them to the surface of the scalp. And really what they use is this kind of so they use it's not a glue, but it's this like sticky kind of grit. So if you've ever been in one of these, now they often use a kind of net that keeps it in place, but back in my day they'd put this sticky gritty stuff in on the electrode, and then put that in your scalp and in your hair, so you pretty much had to go home and take a shower after all of that. And so you're at a particular electrode along the surface of the scalp, but what that is recording is this. It's going to have to sum and amplify those electrical potentials that are coming from the many thousands of brain cells that are living or that are underneath that particular electrode, or around that particular electrode. So what we see, this is just a basic recording over on the right of various stages of sleep, starting with being awake. So our beta waves, when we are awake and busy, that you can see those are really low amplitude, so they're not very high. And they're also a very fast frequency, so how quickly the waves are going up and down is extremely fast. As we get to be awake and calm, we have alpha waves, and you cannot really tell very easily, I think, on this picture, but that frequency is getting lower. And as we are awake and calm, the amplitude is getting a little bit higher, and that you can kind of see. If we go to sleep, we start out in stage 1 sleep, where we have these theta waves, where again, that frequency slows down and the amplitude gets slightly higher. As we get into stage 2, we still see these theta waves, that same kind of speed and amplitude, but we also see what's being kind of shown there in the left, kind of mid-left, where it's really busy, these sleep spindles. And then on the far right, as marked as the K complex, where we see this clear inhibition. We'll talk about this when we talk about wake-sleep cycles. As we move into slow wave sleep, so stages 3 and 4, we start to see more and more delta waves from stage 3 to stage 4, where we see these very high amplitude, low frequency electrical signals, which suggest that the neurons are all communicating at once, and then they're all not communicating for a little while. So a real synchrony, and this appears to be the time when the brain is really resting. And then, of course, as we're going to get into with wake and sleep, we actually go back up from stage 3 to 4 to 3 to 2, and then to REM sleep. So we don't keep going back into stage 1, but then we go to REM sleep, and that starts to look more like beta waves, like we are awake and busy as we get those really high frequency, low amplitude waves. But again, we'll go through all of those. I just think the sleep cycles are very neat. And again, we can tell this from the EEG. And we can use the EEG to get these evoked potentials, sometimes called evoked responses or event-related potentials. And I can't remember what he uses in your book, but from reading in the language field, we talked about them as ERPs, as event-related potentials, where we're using the electroencephalograph, but we're measuring the momentary changes in that electrical activity in the response to a particular event or a particular stimulus that we are interested in the brain's response to. These are really nice because we can look at this millisecond by millisecond change, which again for language is pretty exciting because language happens really quickly. Right now I'm talking quite quickly, and you are getting new words every 200 milliseconds or so, 250 to 300 milliseconds, and you're trying to put that into your representation of what you're understanding of what I'm saying. So if we look at, so this was actually a study where the person was connected to the electroencephalograph, and the words would come in the center. So they were reading the text, but the words would have to come in the center of the screen because when you make eye movements, it causes quite an electrical potential, and that would get in the way of seeing these results. Where if we look on the left, we see what's called an N400, which is a negative going waveform at 400 milliseconds in response to a semantic anomaly. So something that doesn't make sense. Okay, it does make semantic sense. So the cats won't eat is the blue line, and I'm going to call that the normal line. So I'm reading along, and the cats won't eat comes in the center, continues on. We look fine. The cats won't bake. When we get to that word bake in the center, that's my 0 millisecond spot. And you can see that at 400, about 400 milliseconds out, we have this negative going waveform. That we're basically going, that doesn't make sense. Cats won't bake. Over on the right, we're looking at a positive going waveform at 600 milliseconds in response to a syntactic anomaly, so a structural problem with the sentence. So the cats won't eat. You can see that same waveform in blue that you see over on the left. If we compare that to coming up again in the center, the cats won't eating. When we get this syntactic or structural anomaly, we see this positive going waveform out at 600 milliseconds out. My memory is this was from Marta Kutas and her colleagues. And one of the things you will notice is that negative is up and the positive is down. For whatever reason, the ERP literature has negative up and positive down. Just so you know, historically they switched for a little while so that positive was up or negative and negative was down, or at least some people switched, and so it's a very confusing, I think, literature. And you just really have to read and pay attention to both the reading and the figures and how they're going together really quite closely, because they go back and forth just a little bit. But that's a basic, nice ERP result. More recently, they've started using magnetoencephalography, or MEG, which is similar to the EEG, except instead of those electrical potentials, we are reading the faint magnetic fields that are generated by brain activity. This allows, as far as my understanding, it allows a better topography of what exactly is happening where in the brain, as opposed to even those ERP or EEG caps that had, I think the last one that I had any experience with had 128 channels, that there was still, it wasn't as easy to tell exact locations, whereas with the MEG, they can identify approximate locations to within about a centimeter. And again, we're looking at millisecond by millisecond changes so that we really can look at what's happening in the brain very quickly in response to something like language. But in this case, so I like that they use this kind of simple stimulus and a very short time lag. So this is showing the brain's response to a brief tone in the right ear, and this is just 200 milliseconds out from the tone. So this is just kind of processing a brief tone. And you can see, if you look over at the right, just kind of above or around the right ear, there is some activity there. If you move through over to the left, sort of what I'm gonna say is over the temporal lobes, because we're in the auditory, we're looking at the primary auditory cortex basically. But on the left, you see there are a few other places, right? There's a little bit more activity in those places that are around the primary auditory cortex on the left side. And that, if you remember me saying early on, well, now it's just been a couple classes ago or a couple slideshows ago, of how sound is mostly contralateral, but it's really quite complicated because some of it is going up ipsilaterally. You can really tell this in how the MEG, or the MEG, how it is showing our response to a tone in the right ear. I'm going to continue talking about recording brain activity and making assumptions from what we can see in recorded brain activity by talking about positron emission tomography, or PET studies. We are again examining brain function, but now we can map the activity of the brain over time. And they do this by using radioactively tagged chemicals. So basically they have, usually it's something like 2-deoxyglucose, which is going to serve as a marker of blood flow or metabolic activity, as that something that's acting like sugar is going to move into those areas where neurons are active. And because it's been radioactively tagged, that radioactive radioactive atoms are going to decay. And as that happens, they're going to release a positron. It's going to collide with a nearby electron. Those, as they collide, are going to send off these gamma rays in opposite directions. And so we have these gamma ray detectors that can tell where that collision took place, and thus where that brain activity is. And so this provides us a nice color-coded map indicating which of the areas of the brain are active in response to some particular task that we're giving people. And we stopped here with the Peterson et al. Study from 1988, now a really classic study, where they had subjects perform several different tasks, and we're comparing looking at words, to when they're listening to words, to when they're saying words out loud, either read or heard, or when they are thinking of words. Okay, so in this case, a word association task. If they see apple, they might think about orange. Okay, so when they're looking at words, we're looking at the left hemisphere, so the front of the brain is to your left. So this is the back of the brain. When they're just looking at words, we see activity in the occipital lobe, which makes sense for visual processing. And as we are listening to words, you can see that the activity here is in the temporal lobes, the auditory cortex, probably here to near Wernicke's area as well, as we're listening to words, some of those auditory association areas. When we're speaking words, we see more activity there at what looks like probably right around Broca's area as they are saying the words, that motor area for when we speak. And when they are thinking about words or thinking about a word associations, there really is activity over a great deal of the brain. I'm going to say this with words on a slide, but just so you can see, we get this map, this color-coded map of where activity is, but you can see that it's also it does not have good spatial resolution. We don't have a good indication of exactly where that activity is. It's just kind of this general area, and I'm making those assumptions about where that is. It's been a long time since I've read this paper, so based on what I'm assuming is going on with looking at, listening, and speaking, but it's difficult to tell. I'm going to use the example of listening to words. It's difficult to tell that that's exactly where exactly in the temporal lobes that is. And I started with that because PET has some drawbacks. One is that it's slow. Okay, experiments typically take 2 hours. As we're looking at that metabolic activity, and we need that radioactive chemical to decay, that each condition and each trial takes some amount of time. So they are usually pretty slow experiments. There's this low resolution, not just spatially as I was showing, but temporally. Again, we need that metabolic activity. Takes time, and then it's expensive. There needs to be a nearby source of radiation. And it says there are risks associated with a radioactive tracer, and that's not really true. I mean, it's extremely small and it's not really any risk, but people don't like to hear that they're going to be injected with a radioactive tracer. And so people would sometimes balk and say, I don't think I want to do this after they had already volunteered. So as most of us know, the functional magnetic resonance imaging has really taken the place of PET a great deal. It is much more common these days. If you notice, that PET study was conducted back in 1988, that was quite a while ago. And when we were just starting to figure out how to use this MRI machine to look at the functionality, to look at the brain activity. The magnetic resonance imager itself is using magnetic fields, radio waves, and computerized enhancement to map out brain structures. And we'll talk about this more when we're talking about looking at the structure of a living human brain. We use this variation of the MRI. The functional MRI is measuring blood flow and oxygen consumption in the brain to show us where we have high activity. It's sometimes called BOLD, so blood oxygen level dependent imaging. And so what's happening is hemoglobin, our blood, has a ferrous component to it. So it lines up in a certain way in the magnet, like the little tiny magnets within that. Those people are surrounded by a magnetic field. And then when the neurons use the oxygen because they're active, that hemoglobin lines up differently. And the machine can tell us where those differences are to say this is where the brain is active. So as I said, the fMRI is preferred to PET. Like PET scans, we can map brain activity over time, but with vastly greater spatial resolution. So within 1 to 2 millimeters, you're going to see some pictures and you're going to see that they are much more precise in where we're talking about where brain activity is, and greater temporal resolution as well, so greater resolution in time. And I'm not sure exactly how long the PET takes for a kind of trial or task, but within about a second, we're going to see that metabolic activity. And as I was saying earlier, for EEG, or really the ERP, the event-related potentials using EEG, as well as the magnetoencephalography, the MEG, that, remember, those were giving us the temporal resolution of milliseconds. And so milliseconds are a thousandth of a second. And for me, somebody studying language, that's really important. I'm speaking really quickly here, and you're responding to my words within milliseconds. We usually spend, I think the average is something like 250 to 300 milliseconds on a word when we're reading. So a second isn't going to tell me about necessarily natural processing. And language isn't the only example. There are lots of tasks and things that we process really quite quickly. And but this gives us within about a second, which is better than the PET scans. Here I'm showing the MRI machine, so how we would also get information, the fMRI and the DTI, the diffusion tensor imaging. And usually I stop here and see if anybody has any experiences that they want to share with being in an MRI, or doing an fMRI study or anything. So if you want to share something, you can do that on Monday. I won't probably remember to ask if anybody wants to do that, but please volunteer any personal experience kind of information that you want to. So I'm showing these areas of the brain, and again you can see on the top in the top in A, that brain is the red, is showing you activity, and the bottom is the B, is the yellow. They usually do either red or yellow. I think it's usually yellow is the highest activity, and then red, then orange and red, and then the green and blue is the low activity. And you can see how precise, and especially if you kind of go back and imagine that PET study of what we were looking at, this is much more precise, and we can look at these really small brain regions. This picture is showing the parahippocampal place area that is activated by the top row, so those places, but not to the bottom row next to it. So jet planes, jumbles, and things do not activate it, but anything even suggesting a place. So on the far right, those to me look like Legos or something that are suggesting potentially navigating through here. The one next to that is an empty corner of a room. So yeah, even anything suggesting a place activates this area. And then on the bottom in B is the showing the extrastriate body area. If you look at the top row, that's what activates it, and it's any part of the body, a whole body, even a stick figure or a shadow of what looks like a human figure. But the bottom row, all of that, even the face, right? Even though we can think of that, of course, it's part of the body, but we have that fusiform face area that is processing faces, and so that doesn't belong in the extrastriate body area. But again, what I'm showing here is the precision of where that activity is. And your author addresses the subtraction logic, I think, here after both PET and fMRI, and I'm going to use his pictures as well. But you know, we've gone over this for PET, that we are going to have an experimental task, but the brain is always active. So we really want to see what's the activity that's due to this specific task, and so we have to have some kind of comparison task, and we're going to subtract that comparison task, the brain activity to the comparison task, from the brain activity to the experimental task. I think this example was looking at words was the experimental task, although I'm pretty sure the example's made up, but he gives us an example of a possible subtraction. So my experimental task might be looking at words on a screen and reading. And then my comparison task might be something like looking at a foreign language that we know the person doesn't understand. So they're looking at something that's very word-like. The visual, what's going on visually is similar, but the component of reading has been taken out. And what I'm assuming he's really showing there in the difference with the +5 and really the +2 behind it is it's those left areas that are near Wernicke's. He looks like he's showing Broca's area, but we have Broca's area active often when we are even reading words in that this embodiment idea of what part, even though we're not moving our lips and making those motor movements, our motor area is active. So we see that activity in the left, but you can see along the left hemisphere, we're going to see that activity in those language processing areas. And we're subtracting out the comparison task, so we're not really seeing the activity that's in the visual cortex and all of the other activity that's just due to looking at something that's word-like. And traditionally, and how we think of the fMRI is to look at the functions of the brain. So what part of the brain is active when we're performing a particular perceptual or cognitive task? What functions does it play? But it also provides other valuable psychological information. So he gives a couple examples here of when people take a placebo, the regions that are responsible for experiencing pain decrease their response. So one of the things we've learned is that the placebo isn't just, and I'm going to put just a psychological effect, right, as we get to this mind-body problem. It is a change in brain activity. Also, we can predict what people are imagining, or we can predict their kind of intentions. And I say this that we can do this with that enthusiastic cooperation. And I'm going to go show an example with pictures. Okay, so here's an example, and it's a really simplistic example of what people are actually looking at, but we can look at imagining a particular scene, and this can get much more complicated. So in this one, the control stimulus, we're showing them these bars of different orientation, and we're looking at the stimulus. In this case, they're looking at a particular oblique line, and we are measuring from 500 voxels. Okay, so these are like pixels in the brain, but three-dimensional, right? They're voxels in the brain. 500 voxels from the visual cortex, and we're seeing a certain pattern of activity as we're showing them different levels of oblique. The example down in B, the stimulus is horizontal. We can do vertical and all around. So we're looking at the pattern of activity to these different stimuli, and then we can, after a number of trials, many trials of them, again, enthusiastic cooperation of them looking at these, we can put up a stimulus and we can make a prediction. We can say, oh, now you're looking at horizontal lines. And they'll say, yes, I'm looking at horizontal lines based on that pattern of activity in that area of the visual cortex. And while this is a really simple grading pattern, I've seen where they've done this with much more complicated stimuli of pictures of mountains and so naturalistic pictures, but we can use this to make predictions about what people are seeing, imaging, intentions, and thinking about. Again, with their enthusiastic cooperation, they're running through many trials where they're telling us, this is what I'm experiencing.